Today we will talk about on citrate test. So this citrate test is among a suite of in vitro tests, that is indomithyl red phosphorus and citrate, and it is one of the biochemical tests that can be used to identify gram-negative bacilli in the family Enterobacteriaceae. So excuse me. The principle of this citrate test is this citrate test is used to test an organism ability to utilize citrate as a sole source of energy. And the way citrate work is that some bacteria can use a compound called citrate in absence of any other source of carbon. So that means even if there is no glucose, sucrose, lactose, or no other source of carbon for them to use, only citrate they can use as a sole source of energy. And this as carbon is needed to make things like DNA, RNA, protein, lipids, and carbohydrate, and so they can use in absence of carbon. But when other citrate negative bacteria will not be able to do so, because the reason is that they can use citrate as a sole source of carbon is because they produce an enzyme citrate to transport citrate into the cell where they can break down. So um, this is the principle of the citrate coming to the media used in citrate utilization test. For the citrate utilization test, it is performed by using a Simon citrate agar which contain the sodium citrate, then some macronutrients as well as the pH indicator, which helps which is which a color change to tell us whether the test organism is citrate positive or negative, and the ionized water. Now, coming to the procedure, firstly we have to streak the slant back and forth with a light inoculum peak from the center of a well isolated colonies, and make sure that you sterilize your inoculum after streaking the slant back and forth. Then you have to incubate aerobically at 35 to 37 degrees centigrade for up to 4 to 7 days. Now you have to observe a color change from green to blue along the slant. And the interpretation, here in the interpretation you can see in the given picture itself, here the positive turns into the blue color, whereas the negative citrate they remain green in color. This is because when it shows uh, blue in color, it means that the organism has utilized citrate as a sole source of carbon. That is why the pH has changed the color of the test organism from green to blue. Whereas in the negative citrate, here there is no color changes as compared to the positive. So that means no color change, meaning pH did not change because organism cannot utilize sodium citrate to get its carbon. So this is the interpretation of citrate test, and this is also very convenient way to carry out. And of course there are some limitations of citrate utilization, which is like some organisms they are capable of growth on citrate and they do not produce a color change, so growth is considered as positive citrate utilization test even in the absence of a color change. Then this test with equivocal results should be repeated, and the reaction of the medium along are not sufficient for the identification to the species level. So this is all about the citrate test.